you're a teacher, and I wanted to ask you if you see a difference between young adults back then and now. You see, okay. And could you elaborate on that?
I haven't really noticed a big difference. If anything, I find the young people in my group always inspire me. I especially love how tech savvy some of them are. You know, I have a student who is, you have to be careful if you mention the name of a protein in his presence. He will then spend the next, 8 hours doing AlphaFold on every homolog of that thing. No, it's really fun. I love how, and I can't write code and do a lot of the stuff that my students can do. I find it really, really fun to interact with people who are, so grew up with such different experiences from me, This technology that our kids are immersed in, it's kind of scary, but my impression is that the ones, at least the ones that make it into the laboratory front door and end up scientists, man, a lot of them are really on fire. They're like really good, and they're using the technology to really enhance their research, and they're great. So I've been energized by interactions with my students now. They've often always been really good though in the past, so I haven't really noticed how much difference.
Okay.
Thank you very much. I can also repeat the question if in case they need it for the audio back there.
It's quite related question, but so when you're becoming young PI, you're as good as your employees that you're going to have, essentially. And then when you progress in your career, I think you're even more dependent on those people. So what's your secret? How do you choose the right people? And then, what do you do with those that really didn't make it, or whether you keep going, or whether you cut them off? What's your secret behind it?
I don't have a secret. I think that's managing your interpersonal relationships with your students is really, really hard. And to be a successful scientist, you certainly are not selected for your social skills ever. It's like, so I grew up in a family with 3 boys, and we were all like always teasing each other, and when you're a PI, you can't joke around all the time and say things that are that might be offensive or taken the wrong way. It was a learning curve for me. I think I got better over time at being sensitive to my students' feelings, and one of the things that I often find really difficult to deal with is how attached people become to something in the laboratory that they're working on. Like I always think of it as a team effort and as the PI, but I remember being a student too where you felt very defensive of somebody working on something similar to you. But in order to get synergy in your lab, your students have to be able to work together, right? They have to be able to team up. And I often found that I'd be pushing students to work together, and they'd not want to. They wouldn't want to share. Though that's my gene. You can't work on that. Well, unfortunately, that gene interacts with another gene. It's like they function together. So whether you like it or not, you're working together. Because ultimately, you have to. Managing things like that has always been challenging, and it's still challenging. But in the end, it works out, everybody, hopefully. I always try when I'm choosing people, whether to be in my lab, I really like to be able to have a conversation and be able to, have fun talking to the person. That's a really good sign to me, because if you can't have that kind of a good conversation with a person, even someone you just met, then probably you're not a good match personality-wise. So I try to do that. But some people are really quiet or shy, and so you have to give them a chance. And we have this process in the US of rotations, but we don't have that for postdocs, but for grad students we have that. And really works well to help build a relationship, give time to the student to get comfortable and get to know you, because a lot of students are scared to interact or too shy because they don't know anything yet in your field, right? So it can be a real challenge. But some of those students, flourish later if you give them time.
Your co-laureate, Andy Fire, seemed quite shy and quiet, but you got on famously, so...
Yeah. Oh, well, Andy, he's a very socially engaging person. I mean, he's so full of curiosity and fun to talk to. I mean, it's funny because we interacted before there really was much of an internet. And at one point I was like looking, people asked me, do you have like records of your conversations or your emails? And I'm like, I don't think we were emailing much back then. We would talk on the phone, and Andy would talk so much, I'd have to like switch ears, right? My ear would get sore on this side, and I'd have to switch the phone over here. This was when you didn't have a cell phone. You had to hold this thing like this. It's a big clunky thing that's on your desk. It's funny because kids don't even know that used to exist. I still have a phone like that on my desk, and I hardly ever use it. Anyway, even though he's very quiet, once you get to know him, he's a very socially engaging person and full of ideas. And that was one of the great things about my hard time in graduate school when things weren't working. That's when I got to know Andy, because he was working on DNA transformation in C. elegans independently of me, and we got to know each other because it was hard work to get it to work. We didn't, he made some breakthroughs and I made some, and together we really got it working. But that's a great example. But as a colleague, we didn't have, like, at least initially, I think there was a sort of a standoffish thing. We didn't hit it off right away, but we did gradually over time. And maybe that's what you're referring to. Once you get to know him, he's such a warm and engaging, thoughtful, great colleague.
Because you said some students are very shy and they don't open up, and also because of the different education background that they've got, because since I'm from India and I'm in Poland, and sometimes I shy out to talk to people because I feel there is some sort of language barrier, or the way that I perform the experiment and the way I want to convey them is quite difficult sometimes. And at the same time, I also wanted to know from your perspective, do you think interracial labs are more balanced than the lab of their own people? And how are professors dealing with it?
Well, one of the great things about the United States that used to be great anyway, making it great again, I'm not so sure. But one of the great things about the United States is it's very mixed in the way that it's been populated by immigration and by so many different nationalities. And for me, as a human being, I do not recognize race as a difference. I just think of us all as humans. I really am not, and I'm not nationalistic at all. I think it's wonderful that science is a sort of a global enterprise, and that especially biomedical science, we freely publish, we share our data, and it really is a worldwide enterprise. And in our labs, until this recent change in our administration, we've benefited so tremendously from people, not only immigrants, but also just visitors who come on visas to work in our laboratories. It's been wonderful for science in the United States. Science in the United States used to be great. It's not great now. We are in peril. It's scary times. It's existential times. I really don't know if I have a job next year. It depends. Yeah, I will. You guys are wonderful, and boy, do you have a lot of brain power here. I think it's wonderful, the enthusiasm in your people for science. It's impressive. And so, yes, this is a serious challenge right now. What made the United States the leader in biotech and biomedical research were people from all over the world who we welcomed to our laboratories. Many of my students are from China. I have like 5 or 6 from China right now. They don't feel welcome anymore. A lot of them are scared. They don't feel like they can speak freely anymore. And they can't. I don't even know if I can speak freely anymore, sadly. But I'm going to anyway, you know. But I do think we could lose our entire research enterprise. At least a lot of damage has already happened, and it will probably continue for the next couple of years. But we'll see. It's challenging times. Really wonderful to be here, and though to see that there's hope, and I'm very hopeful, seeing what you people here are capable of, and science will go on.
So I really love what you said during the lecture, that we should actually sometimes go for the complicated answers to the questions, right? Not always for something simple. But from another side, I mean, how to convince or help to our people in the labs to actually be persistent on these questions and not to get lost in them. So this is something I'm struggling sometimes.
Yeah, I gather you're a PI running your own lab as well. Yeah, it's a challenge. That's another reason I like it when people team up. A lot of times, you know, and I have to say, the most successful students that I have had in my lab have been the ones who collaborate the most, who work together. They publish a lot. I had one postdoc who was on 12 papers from my lab in the course of like 4 years, and he went on to an independent faculty position where he's doing fine. But in my lab, he was just so willing. He's such a nice person, and he'd help anybody. And he was just really good at what he could do, and he was so successful. Then I had another student who was like that too, and they just sought people out and helped them. And then I have other people who are really independent and protective of what they work on, and they don't want to share. They get defensive, and sometimes they do brilliant things. Sometimes usually they don't produce as much, though, you know. So I do try to work on my students to get them to be more collaborative, but I also, you know, I often push these complicated problems at them too, you know, and I'm guilty of trying to steer them towards, well, you know, what about this really weird thing? And sometimes they take it and run with it, and other times they fight back and push back, and they don't want to do it. Maybe they're right, maybe I'm right, I don't know.
Or they actually asking, for example, why are you branching again there?
Yeah.
It's a discrete sample set, and it's a bit skewed, but if you take Nobel laureates in the sciences, I mean, you've been a Nobel laureate now for a couple of decades, so you've met an awful lot of them. I've met a lot of them, and I would have the general observation that they seem to be quite nice people. And it sounds a bit silly to say, but I think it connects with what you just said about the success of sharing and just wanting to talk to people and be communicative and give your data. Would you agree with that about your fellow Nobel laureates?
Notable exceptions. I won't name any names. No, I think Nobel laureates are just people. I mean, we're full of flaws, but in general, I think scientists are super nice people. Really, they are. There's something about being a scientist that makes you more humble. And some of the scientists I know who are the least nice people, I won't say they're bad people, but they're not friendly. Some of them have, I think, been too successful, too young. It's like they didn't have the humbling experiences that really would make them more, down-to-earth and nice people. I really do think that sometimes that's true. There's something really humbling about doing science, especially when you constantly, like I experience, have an idea that proves to be wrong, and you have to move on. You learn something new and you move on. So I do think that brings out the best in people who stick with it. So I do think being a scientist is a way of continuing to grow as a person constantly, right? I mean, what is science if not the continuous effort to learn more, to make the world a better place? So I do think that brings out the best in people to be a scientist and to be a successful scientist. I think there has to be some sort of a completeness to your person, your personality, something that drives you and makes you always curious and always willing to grow, and really importantly, always humble and willing to admit you're wrong. It's such an amazing thing. If only our politicians would come out and say, "Oh, I was wrong about that." It's like,
yeah, I was also trying to get to this idea of of really sharing more than you take. And it's such a difficult thing because you have to hang on to your best ideas, perhaps, otherwise people might steal them. But at the same time, it seems to me that the Nobel laureates as a sample set tend to have given away a great deal, and what they've reaped back in return has been greater, and it's allowed them to make more progress. But I don't know.
No, I agree. Sharing your ideas is the best way. The best thing you can do as a scientist is to share your ideas as freely as possible, in my experience, because I've always, you've probably noticed, I'm enthusiastic about how cool the things are that we work on. If I do anything well, is I convince other smart people like Andrew Fire that this is a cool thing. Isn't it amazing, this thing the worms are doing? I got him excited about it. I interacted with him, and I told Adam this, I published it myself. He thought of the double-stranded RNA might be the trigger. I was all about the cool phenomenon and how it must be amplified, and I wasn't thinking, how is it triggered? How is it initiated? And he's like, maybe it's the double-stranded RNA nature of the initial trigger that was like triggering this response. It was clearly engaging some sort of an amplification mechanism. And so if either strand could trigger it, the antisense strand could trigger replication of some kind, like an RDRP, then you didn't need, the initial stimulus didn't need to be double-stranded, but he had that insight. And the only reason I was able to be recognized for this double-stranded RNA, the Nobel Prize was for double-stranded RNA as a trigger for gene silencing, was because I got Andy excited about I won the Nobel Prize because I got Andy excited about RNA silencing. I helped later on the genetics that found the Argonauts and the connection to transposons, that came out of my research. So I'm not feeling like a total charlatan, but still, by sharing your ideas, putting yourself out there on the line, your idea might be stupid. Seriously, you could be so wrong. and be embarrassed. And I've had that happen to me, not too many times, but enough times but on the plus side, putting your ideas out there is really a great way to find, to test them out and to have other people get their feedback. Sometimes they'll tell you, hey, I'm working on something similar. Like if you work, I'm sure all of you do this, but talking about unpublished results at a meeting, I've had so many really positive interactions by doing that. Whereas if you ever keep it a secret, don't talk about it, now you're really running a risky business, right? Because then if somebody else is working on it, they're not going to tell you because they don't know you're doing it. So it's really interesting psychologically, but my experience is openness will make you more successful, and so I would certainly encourage that, and I try to live that way. Sometimes like Adam was saying, I really, an idea that I didn't talk about today in my lecture or didn't come out in the interaction is this new concept of RNA interacting, like the non-coding part of the genome interacting with the coding part in what I'm beginning to think of as a neural interface. It sounds really cool. I think it really is cool, and conceptually I think it can explain a lot, but to explain to you why I think this is happening, I would have to go into all these little details. I tried to do this yesterday with the team was interviewing me. It was really funny. We were doing the interview with, Adam wasn't even in the room, and the question came up about this neural interface idea. Did I do an okay job in the end?
I felt like it wasn't complicated when you said it, but there's no way I know what it is. But it was a good job.
But
I tried.
Saffron came out afterwards, and she said he was really worried that he wasn't getting it across, but I understood it. So you must have done a good job.
Yeah, it's really cool. The concept that like I didn't even know how a neural interface works, but AIs learn by forming like the equivalent of synapses or nodes or whatever. I learned this from Jeffrey Hinton's talk at the Nobel Prize this year, where I got to go to because Victor got the prize and he's in my field. Anyway, I was so inspired by Jeffrey's talk about how AIs learn, and I was thinking maybe that explains this weird fact that RNAs, for example, they regulate transposons, but they don't match the transposons. Like they use microRNA like sort of pairing with their targets, so they're not perfectly matched, but they regulate them. Anyway, the bottom line is I got this inspiration from thinking about this more, that the interactions that regulate the transposons are like a neural interface, and that the base pairing interactions are kind of like synapses that are strengthened or weakened over time, but because multiple RNAs are engaging one target, they don't actually end up conserved. The RNAs are functioning collectively to regulate a target, and so over evolutionary time, the idea is that there'll be RNAs that there'll always be RNAs targeting that gene, but different RNAs will target that gene over evolutionary time. And actually thinking that through, we came up with an informatics test of that. And so now we're trying to test this idea, and the prediction is that for certain genes that are regulated by RNAs, they'll still be regulated by RNAs in related species or wild isolates, but it'll be different sets of RNAs, because over evolutionary time, the RNAs that engage that particular target will change, but if it's beneficial for that sort of synapse to occur, it will be selected by natural selection. Since multiple RNAs target any given gene, multiple RNAs will always target that gene, but they'll always be different, because it's kind of like the interactions are like this, the interface, like when an AI learns, it's not exactly the same connections that create the learning, the knowledge, the nodes, the memories, if you will. But the fact that, anyway, I'm probably totally wrong about how AIs work.
I think what you're doing is seeding an idea. I think we need an evening of you and Jeff Hinton together.
That would be fun.
That would be very nice. I think we can organise that.
I actually sat next to him at the dinner.
You did?
Yeah, what a wonderful guy, and such a sad story. You probably know the story. His wife died before the prize, and she was his biggest fan, and really would have loved to be there for the prize, but sadly passed away just the year before the prize. And I don't think she ever knew whether maybe for many years there was speculation he might get the prize, and he was sitting there at dinner, and that came up in our conversation that he wished his wife could have been there. What a wonderful man, but also very great speaker. If you haven't seen it, you can go on to the nobelprize.org and click on his talk about AI. It's really beautiful. Did you know AIs need to sleep? Yeah. It's like you give them data and you let them assimilate the data. You show them pictures of cats and pictures of dogs, and you teach them this for a very long time. But then at some point you have to let them process in the absence of input. He calls that sleep in his talk. And that's because I guess like the nodes that form when it's sleeping are noise, and it needs to be able to recognize signal from noise. And part of this ability to do that requires the system to be sort of firing in the absence of input. And he speculates in his talk that this is why the human brain needs to sleep, which is really interesting, because maybe we're learning about how our own intelligence works from artificial intelligence. But I think this principle of a neural interface storing information in biological systems is really cool. I think it's really cool. And my mom thinks it's really cool. She's 89, and she's like, Craig, you gotta publish that. That is so cool. And I'm like, Mom, we haven't even tested this idea. Believe it or not, I got my mom excited about this neural interface idea. It was so hard to explain this to my mom. It's like, but eventually I felt like, oh, she gets it.
Did you mention it to Jeff Hinton at dinner, or did you?
No, I hadn't thought of it yet.
Okay, cut you off. Right, okay.
It took quite a while for it to sink in. I'm going to hate it so much if it turns out not to be true. But we actually have a test of it, so we're doing it right now.
More questions? Do you want to ask another one from the back? You've got your list.
Yes, I can maybe. I don't know if it's the last one or no, but I wanted to know if you are aware of who you are, like a great figure in the scientific world. Like, is it hard for you to understand like what you did, and how do you feel?
I don't think of myself. I definitely don't think of myself as a great figure. I mean, I keep talking about the great things about the United States. I mean, and it's probably still true. I mean, people don't respect Nobel laureate, who cares what scientist, like you're just a nobody. It's funny though, no, it's true. It's true, like we're very unappreciated in the United States, maybe deservedly so. Maybe it's our fault, I don't know. But there are people in the United States who are very appreciative of but you don't go around I don't get treated like this in the United States. I think it's wonderful. Maybe you do a good job. Even in the United States, Adam can get a crowd together who's great. But I do think that the enthusiasm for science that you guys have, I mean, I'm enthusiastic, but I'm just resonating what I see in the room. Like today, I really enjoyed talking to people. I think I got through 10 slides. And Adam knows, in Lund, I gave a horrible seminar. I went through all this data. It was terrible. I lost the audience. But from you guys, I got so much energy looking out into the audience and seeing people smile. I love to do that kind of a talk because I know that I'm connecting with people, and that's fun. But no, I don't feel like I just feel very fortunate to have had the opportunity to learn so much from this amazing animal we've been studying, and getting to share that with you. It's like, I remember, you know, I like to sail, and one time I did something really crazy. I went on a sailing trip for multiple days with my Hobie cat. It's a small sailing catamaran, has no other source of power, just wind. And I brought my tent and camping gear and some food, and I sailed from Rhode Island to Long Island in New York. And along the way, I saw a great white shark. It swam right at my boat. It swam the dorsal fin and everything, the big eye and the teeth and the whole thing. And I was like right where they filmed Jaws. I was like, really, I was at this place called Montauk, and this great white shark takes an interest in my boat. He swims right up to it, and then I have to swerve. This is an animal that could eat me, and it goes right by the stern of my boat, 6 inches from my rudders, and I am sitting 6 inches out of the water. I have a little seal nugget in my wetsuit that it could have chewed up. Anyway, funny thing is that during the end of that sailing day, I came to shore and I had to find a place to camp, and it was almost sunset, so I just navigated into the coastline, and I came into this little inlet, and there were kids playing on the beach. It was a beautiful evening. And I pull up onto the sand, and the parents walk over, and I'm like, "Can I camp here on your beach?" And the kids are all jumping on the boat's trampoline, and they're like, "Yeah, sure." And I say, "Well, that's so awesome." And I say, "You know what? I saw a great white shark today." I feel like I get to tell you guys about the great white shark. I'm telling you stories about my adventures. I just feel so thrilled to be able to share the adventures that are part of my life, and it was so fun to have the little audience of a few kids. "You saw a great white shark? That's so cool." And I said, "Yeah," and I was only a half a mile from a beach with thousands of people in the water. And sure enough, the next day, someone caught the biggest great white shark they'd ever caught. There were a bunch of them apparently off of Montauk because there was a dead whale and they were eating the carcass. So I had seen this huge animal. Really, that's what it's like. It's coming back to the fire after a hunting expedition. I just imagine how our ancestors felt to come back and say, "Oh, well, you saw this great big mammoth, and so and so speared it," and I just feel like that's what. It's life is full of adventure, and science should be like that. So getting to share that is so much fun.
That was a really beautiful analogy. That was really lovely. And it reminds me, years ago we did this program where we brought together with a National Geographic Society, Nobel laureates in the sciences and explorers. And you'd sit, there were two people, they'd sit and exchange stories. And again, one of them was exploring with the mind and in the lab, and one of them was exploring the deep ocean or something. But basically, it was exactly what you said. It's just telling stories of what you find out there.
Yeah. It's so fun to have that kind of interaction, So I love that.
Going to this adventure you just mentioned, and you also said during the lecture that there is so much of empty space, right? That, so much to discover. Yet the science is becoming less and less destructive. So what is your grasp on this? Why we are kind of, rolling over and over again around the same stories instead of, going and exploring this empty unknown?
Yeah, I'm not sure I understand your point about how science is changing. Do you have a sense that science is not also still growing and spreading the way it should, or is it?
Something
We are
too trendy.
just rolling around the same ideas instead of discovering, going out of this kind of settled things.
Yeah, I do think that we tend to become complacent and be trendy, and you know, I know that it was great when RNAi was really hot, and then CRISPR got hot, and you know, it's easy to publish when something's super exciting. So a lot of people publish in that area. I think the CRISPR field is still like that. All of the amazing things that the microbial world is doing with guided search is just so cool and amazing. But I don't know if I agree with that it's becoming less. I think it just does this all the time. You know what I mean? I feel like in my own experience, it happened with RNAi, it's happening with CRISPR. I think there's good stuff percolating along that nobody's heard about yet that will become the next trendy thing. You know what I mean? It's how humans work. You know, we tend to all sort of get excited at the same time about something, and then that's all that anyone wants to talk about. But it's not a good thing. I agree with you. You know, diversity of interests and exploring broadly, all of these things are so critical to the future success of the scientific enterprise. And I feel like, you know, that's one of the great things about academic science, that we need to keep it going and keep our young people engaged in. So having in your university a real diversity of different laboratories and research is so critical. And then, but also getting them to interact. One of the things that we did is form this RNA Therapeutics Institute, and some of the absolutely most successful labs are pushing translational medicine, translational research, and a very typical graduate student program would be to come into the lab of one of my colleagues who's in this area, develop a guide RNA that works to target a certain disease, do a few tests in an animal model, show that it maybe has a therapeutic effect in the animal model. It's kind of formulaic. Not to say it's not important, because like they might be breaking open brain delivery, or having broken open brain delivery, be testing out a hypothesis like this ApoE4 target, which is really interesting, Alzheimer's target. Does hitting that, you know, for example, this was a great graduate program, but it's very focused, right? It's very focused on making an application out of a tool that was the result of a discovery, you know, years ago. And so that kind of science is a very different kind of science than the kind that I do typically. I think it's cool. I like it. I'm so glad that people are doing it, but I wouldn't want to have to do it myself. But I really like that people are doing it because it's important. But it starts to get close to what a pharmaceutical company could be doing, you know, which is good. They're going to do it because they can make a drug. So in the academic lab, should you be studying something that, you know, that a pharma company might do anyway for profit? And that's where, for me, it's a personal choice, but again, maybe gets to your point about how people work on things that are kind of not on the cutting edge of the unknown, but rather more applied.
Thank you. I actually had a different question, but it resonates with my question in the lecture. Like, you know, if we do basic science and we don't expect the translation, and then it happens, but when we expect to do translation, very often we fail because it's too much pressure. Maybe we are just tunnel vision. I don't know, but this is more of a comment. But because you talked about shark, it's going to be crazy. But anyway, the longest lived vertebrate is Greenland shark. Yes, it lives for more than 300 years or 400 years, and we don't know why, really. You know, but do you think we can use the knowledge that we know about small RNAs RNAi, maybe to control aging in a more sort of holistic approach, not just one gene per time.
Improve health during aging.
Yes, but maybe
yeah,
like
I mean, that's why
sort of
this ApoE4
cocktails.
is such an interesting target, because it came in part from human GWAS, genetic studies of people who age well, people who don't get dementia when they age. They often have a mutation in this ApoE4. And so losing this gene may be good for brain health. I think it also causes like hypercholesteremia or something. It's not good if it's knocked out in the liver. So knocking it out only in the brain might be good. But I think there's a lot of human genetics coming along that suggests targets for improving health span, aging well. I think we're a long ways from immortality, unfortunately. But, but yeah, I'm really excited about using siRNAs to improve the quality of life and aging. So are my colleagues. Of course, you can make a lot of money that way too, but that's the amount of benefit it could have if you could make really impactful drugs that improve the quality of life as you age. So that, and I do think it's possible. I really do. Although you can also just exercise and eat right and improve your longevity that way too.
Now you're getting radical.
Because the way I think, we are so complex, and you're talking about C. elegans with 900 odd cells, but we have billions of cells and 3.2 billions of base pairs, and it's all being transcribed in the same time. So there will be such a complex reaction. Even if you take one siRNA against anything, there will be tons of side effects. And how do you control, in the context of a, experiment with mouse, whatever, you can, but in the context of human life,
yeah.
How do you control for that? Or you just don't. You just simply look for the lowest hanging fruit and you go for it.
Yeah, I didn't talk about this, but siRNAs generally can be tuned to be quite specific so that they don't. You have to use informatics a little bit as you design them to try to avoid seed targeting, and you have to also look for off-targeting. It was a big worry for many years before siRNAs really became successful in the clinic, that there'd be off-targeting and targeting of genes that had partial match to the sequence, and that does happen, so you have to be worried about that. But in principle, it hasn't been so far difficult. To my knowledge, there are no programs that failed because you could not find an siRNA that had sufficient discrimination. Whereas in the case of monoclonals, for example, there are many, many cases where the monoclonal antibody hits a closely related receptor, for example, and so you can't make a clean drug. But with an siRNA, there's even an siRNA candidate now for a rare single nucleotide polymorphism where the wild type function needs to be preserved, but the mutant allele needs to be silenced because it causes a terrible dominant disease. That is a super orphan disease. I'm going to have to learn how to pronounce the FOP, fibroplasia ossificans progressiva, I think is what it's called. We love these jargony terms. What it means is you bruise a muscle, the muscle turns into bone. It's a horrible syndrome. Apparently, it's kind of famous because the skeletons of a patient who has this, they look unearthly. Sadly, it's a devastating illness. It's caused by a mutation in the ACVR1 BMP receptor that makes this master bone signaling pathway receptor responsive to the cytokine activin A, which is secreted. It's also a BMP, but it acts through a different receptor But with this mutant, that bone responsive receptor responds to inflammation signals. And so when you get an injury to a muscle, it turns into bone quickly. Incredible disease. But one of the things that's amazing is it reveals something that most people don't know. Our bodies have an incredible capacity to build bone, muscle, and fat. We all know that it can build fat. That part everybody already knows. But there are a lot of conditions like osteoporosis and arthritis, et cetera, where bone is lost and or cartilage is lost. And our body's ability to make those tissues is really huge, and it's untapped because we don't have drugs that stimulate that. Pharma's been trying for quite a while, but we now have siRNAs that stimulate that. And part of it comes from one of my colleagues who had this terrible FOP mouse model. It has a humanized allele identical to the human lesion, and because this person doesn't work on RNAi, had this model in the lab, it kind of became a challenge for Anastasia Kurova, who I maybe didn't talk about today, but I'd love to talk about her work because she's amazing. Hundreds of patents for her work, and she broke open brain delivery. And so she thought, well, let's try to see if we can make an allele discriminating siRNA, one that can silence the mutant allele but not the wild type allele, because that's what you need for this disease. And so she tried, and they found an siRNA that worked pretty well. It has single mismatch between already, because the mutant, and they put that single mismatch in the seed region, which is a really important region of the microRNA, the first part that engages the target. That's particularly important. So they positioned that, and it wasn't good enough. It still knocked down the wild type allele. But then they walked a second lesion through the siRNA, looking, now the mutant allele mismatches by 2, and the wild type allele mismatches by 2, but the mutant only by 1. And now the discrimination was 100-fold or more. So they developed this siRNA and demonstrated that if you give a dose of siRNA by sub-Q injection into this animal model, this mouse model, and then wait about a week and then injure the muscles, no bone growth. Completely blocks the bone growth. Otherwise, the animals are normal and healthy. I think it's going to work in the human, and it's going to prevent that's an exquisite amount of specificity, that it can have that kind of specificity as a therapy. It opens up many, many doors. So it's really exciting. I think the future of this technology for very precision medicine is really good, really great.
Thank you. Okay, maybe we've got time for like 2 more questions. So if you want to ask something, now's your time. Or I know you have more questions at the back, but...
Go for it.
Yes, I would like to ask, when you were working on your Nature publication, was there a time that you knew that it was something really, really great and that it would be in future part of the Nobel Prize?
Absolutely not. No, there was no inkling. Although, if you had heard me talk about RNAi before the Nature paper was even written, you would think it was going to be, my mom probably thought it was going to be a Nobel Prize. I didn't think it would. The reason it was just purely phenomenological, which to me is very frustrating. When we wrote that paper, the 1998 paper, we had no idea how it worked or what the mechanism, whether it would be conserved or not. I mean, I think there was a lot of good reason to think it wouldn't be. I remember talking to a colleague who works on Drosophila, and he asked me, do you think it'll work in fruit flies? And I said, no, probably not. And the reason I said probably not was because people had been injecting RNA into fruit flies for a long time, and never saw this, right? So, and so it wasn't until people, in part because of the 98 paper, that colleague, actually, he was the first person to get it to work in the fruit fly. Once they made double-stranded RNA and injected it into the fruit fly, it worked beautifully. And you can get a really beautiful copying of the loss of function phenotype by triggering it in the fruit fly. In the past, in the fruit fly, the researchers were always very careful to purify the mRNA and inject the mRNA. And I don't know how much antisense was done, but it was they never saw this response in the fruit fly. Fruit fly doesn't have the RDRP system, so it doesn't amplify quite as much. But if you give it a nice bolus of double-stranded RNA, it beautifully phenocopies it. And that person who I said it wouldn't work was the first person actually to get it to work in the fruit fly, which was a huge, exciting moment. But it only came because he had to make it double-stranded. But for me, the moment when I realized that we had something super, super cool was when we got the transposons mobilized, and when we realized that the gene was a member of a family of highly conserved proteins. It looked like it could be something really, really cool. At that point, I felt pretty sure we were onto something really, really cool. So that was fun.
I can maybe ask one somebody new. Okay. Yeah. I got very simple question. Looking back, if there is anything that you could have done differently in your leadership or your career, yeah, that's the question. Yeah.
That's funny because I was asked that by a reporter. Adam probably knows all my stories now. He didn't know about the shark. That was a new one. But
I got a bad memory. I keep forgetting them anyway. Tell me again.
I was asked that question. You know how RNA interference went through this period of incredible hype, and the Nobel Prize was awarded in 2006 before all of the clinical trials failed. There were a bunch of early clinical trials, and they all failed right around 2007. It's like, talk about great timing, right? If they had just waited one more year, they probably would have decided not to award the prize for double-stranded RNA. But right about, so 2006, we get the prize. 2016, 10 years later, still no drugs. And in fact, very little investment. Most of the companies who were well invested back in 2006 were out of business. I had a company I co-founded with Anastasia as the chief scientific officer. That company failed. We recruited her to UMass, and she's a chemist, and she's like doing all this chemistry to stabilize the siRNA to try to improve it so that it would work. Because the stuff they put into the clinic, it had no chance of working. I knew it. I think the people who were doing it knew it. The investors probably knew it. But sadly, you can still make a lot of money from all the hype, right? So if you have the first RNAi company who says they're going to do something big, and you make an investment and then you go public, investors cash out, they make a 30 times increase in their value, then the drug fails, and oh, too bad. You know, this happens all the time. Very cynical, but true. Right around 10 years later, all this negative stuff, and the reporter called me up and she said, well, it's been 10 years and there are no drugs. Would you have done anything different? Exactly your question. And I said, yes, I would have hired more chemists because it took a lot of chemistry to make this into a drug. And the chemists love it when I tell that story. It really is true. Biology and chemistry work beautifully together. We're just so fortunate to have our chemistry colleagues working on this because the chemistry space around oligonucleotides is huge. Even in nature, RNA is modified in hundreds of different ways. And we understand very little about how those RNA modifications change the behavior of RNA. This led to the Nobel Prize for mRNA because making RNA with pseudouridine for some reason makes it less immunogenic, right? Sort of an empirical discovery, but a really important one from the standpoint of getting a drug out of mRNA. There's a lot more of those sorts of things to be discovered. And of course, there's also in completely bioorthogonal modifications to RNA. One of the ones that's led to this this drug that will go into hopefully these patients with this horrible bone disorder, that probably only works because the RNA backbone, the phosphodiester linkage, has in this siRNA derivative that's incredibly stable and distributes to these peripheral stem cells that have this multipotency. It has a methylene, a carbon group inserted into the phosphodiester linkage. It is completely bioorthogonal. There is no organism on the planet that ever puts a carbon atom into the phosphodiester linkage. It turns out if you put one there chemically, the nucleases have no idea what to do with the 3' end of the oligonucleotide. And yet it's not toxic, it's biodegradable, it still loads onto the search engine protein, RISC. Biological organisms can never make that. They do not make that. They probably could if they wanted to for some reason, but they don't. So it's really that kind of thing that chemists can do. And so absolutely having that's kind of probably not what you were after, but that's an example of one thing I would have done differently. But yeah, I often think about that, how lucky I am to be where I'm at, in my career. And there are many places where had I done things differently, it would have worked out a lot worse. So I'd have to say that on average, I'm pretty happy with my choices. So yeah, it's fun, but definitely hiring more chemists, that's something I should have done earlier.
I guess, what did you expect but a scientist's answer to your question? I mean, after everything you've heard. I think that's, you've worked so hard.
Hey, I love it. I mean, you guys really, you're fun. I just hope you all enjoyed this as much as I did. You're great, and it's really fun to interact with you, and just don't change. Keep it up. You give me hope. You really do. It's been hard times lately for science in the United States, so thanks for being who you are and continued. I hope to be that way. I know you have some elections of your own, so good luck.